Thank you, Dr. Joukowsky, for an inspiring talk. We're here to celebrate the discovery and the development of embryonic stem cells, or ES cells. And this was a single advance that made a universe of discovery possible. They're a miracle to scientists and even more, I think, to non-scientists. So we all know how an embryo develops. A sperm unites with an egg, and the united cell combines its genetic material. So half of the genes come from the mother and half of the genes come to a father. The embryo grows and divides and implants into the uterus and grows and divides and is organized. And this gives rise to the many tissues and organs that ultimately give rise to a complete new organism. But the embryonic stem cell is a completely new way to make an embryo. Now what this is it's a cell or a group of cells that live in a dish in the lab. And there in the laboratory, you feed them and you freeze them and you thaw them and you ignore them and you maltreat them. And then magically, you can take these cells, you can inject them inside the hollow ball of cells that surround a developing embryo and fool the embryo into taking those cells up as part of itself. And a few weeks later, there's a mouse running around in the cage, because this is a mouse that we're talking about, and that mouse's brain and heart and lungs and liver and blood are derived from that unspecialized, uninteresting, unexceptional cell that was growing in a dish. And the thought of this still actually sort of makes me dizzy. It's amazing, just a science. But it's also really important from a biomedical point of view. So we are humans and we are mammals. And in order to understand mammalian biology, there's a certain amount of science that needs to be done on mammals. And a lot of that is done on mice. And the reason is because mice are small and inexpensive and fast breeding and pests, actually. They're vermin. But they are very much like ourselves in their physiology, and the existence of embryonic stem cells makes it possible to generate mice that have diseases that are very similar to important human diseases, and that resemble them really in what causes the disease, and it makes it possible for us to try to develop treatments and understand disease progressions in ways that are ethical and efficient and simply could not be done. So we talk now about mice that have sickle cell anemia, or mice that have cancers related to human cancers, or mice that have diseases of the brain related to Alzheimer's disease. And these are all really important models that we use for study that grow directly out of the development of embryonic stem cells and the work of these three women. So strangely enough, in the prize community, embryonic stem cells have not really received the sort of celebration and appreciation they deserve. And this occurs paradoxically because the development occurred the way science is really supposed to work, which is as a cooperation and a communication and a collaboration between different scientists who all pooled their expertise, who all pooled their ideas, and put it together in something larger than any one of them could do. And the prize-giving system tends to celebrate the one big man with the one great idea, rather than recognizing the way that something organically grows into something important that everyone can share in. And that's been the case with embryonic stem cells as well. And because we have this Pearl Meister Greengard Prize, we can actually pick out 3 women along the way who made absolutely seminal contributions to the development of embryonic stem cells, to all of the work, and celebrate them as part of this community of scientists. So I'm going to start with Beatrice Mintz, who couldn't be with us today, who was the senior member of this group. She graduated from Hunter College in 1941. She was a scientist at a time that there were not a lot of women in science. And she did many creative experiments showing us that the mouse embryo was much more flexible than we had realized that embryos could be. And for example, one of the experiments she did in 1962 was that she showed that she could push 2 mouse embryos together in the right way, and after that, the 2 embryos would fuse and grow up to become a single mouse. So we've just violated the laws of Mendel. Now we have a mouse with 4 parents instead of 2, the 2 different parents of the 2 different starting mice. And Actually, we now know that this is sort of the opposite of what causes an identical twin, a single embryo splitting into two. You can have two embryos fusing into one. And this actually happens very rarely, but occasionally, even, and they're normal humans who run around who have four parents, or who started from two embryos instead of one. And these people can be fine. So, but this tells us something. It tells us that the embryo is it's flexible. It can be put together in different ways. And about 10 years later, Beatrice was studying a kind of tumor that reminded her of a disorganized embryo. It was something called an embryonic carcinoma. And within this carcinoma, you could find muscles and bones and blood and all sorts of strange tissues. And she thought about these embryos, and she remembered the ways that you could put two mice together, and she thought, I wonder if these tumors, if they were put together with another embryo, could combine and make a normal mouse. And this actually sounds completely crazy, even looking at it now, but it worked. So when put into a normal mouse, these tumor cells were organized by the normal mouse, they were instructed as to what to do, and they could give rise to the bones or the brain or the blood or parts of this normal mouse. They could contribute to the mouse. And she came up with a very clever word for this. She called these animals chimeras, based on the creature from Greek mythology that has the head of a goat and the body of the lion and the tail of a snake. Now, these animals were all mouse all the time, and they didn't have all these different parts, but they did represent a fusion that never existed before it was made in a laboratory. And the thing that's important about this transition from cells to an animal is that it makes it possible to, we can manipulate cells in the lab. We can manipulate them in ways that are not possible in a whole animal. But they were limited because there's only so much that cells in the lab can do. And the ability to go back and forth to create an animal, a mouse, with all the different organs and physiology and experience that an animal can go through means that we can have it all. We can have the power of the cells in the dish, and we can have the complexity of the entire animal. Now, this is a great start, but actually there was a problem with embryonic carcinoma cells. They could sort of contribute to a mouse, but they couldn't make a whole mouse. They were cancer cells, they were abnormal. There was sort of a partial solution to the problem. And I think of Beatrice Mintz in some ways as like, we're coming on Passover here. She's like Moses, who was able to see the promised land, but was not able to go there herself. She was not able to reach the next step. And the two women who made that next step and who brought us into this new world of biomedical possibility are Gail Martin and Liz Robertson. And I think it's a really great use of the prize, and the committee felt that it was a wonderful use of the prize to point out that we aren't just honoring one person at the end of a distinguished career. Well, we're also honoring the contributions that young scientists coming out brought in about how new people could build on what had been done in the past and generate something new that hadn't been built before them. And so Gail Martin took the next step in this important advance as when she was just starting out on her own, and she knew about these embryonic carcinoma cells, and she learned a way to create similar cells, but starting from a completely normal embryo. She learned how to turn cells from a normal mouse embryo into these cells that you could grow indefinitely, that would be sort of undifferentiated and unspecialized and subject to laboratory abuse, but that could then be combined with an embryo and could give rise to all of the different cell types that are in an embryo. And in fact, the word for that possible they have is that they're totipotent, they're capable of everything. And Gail demonstrated this. And she also coined the term embryonic stem cells, which is the word that we use to describe them today. So this tells us that we could really go into normal cells. And then Liz Robertson, who was working with Alan Bradley and Martin Evans, who had independently isolated ES cells, took the next step and the critical step to make the whole package work, which is she showed that these cells didn't just contribute to the first mouse. These cells could then go into the germline of that mouse and make whole generations of new mice. From these cells in a dish, you could generate not just one abnormal animal, but whole genetic strains of vermin that had been at some point just cells in a laboratory, what seemed like sterile life, but are now whole strains of animals. So the scientific conversation among these three women and among other very distinguished and extraordinary men and women has really revolutionized biology. In 1977, Gail and Beatrice worked together to make the first mouse engineered to have a mutation analogous to a human disease. In the mid-1980s, Oliver Smithies and Mario Capecchi developed innovative ways to change the DNA of embryonic stem cells. And in 1989, Liz Robertson was one of the very first to generate a mouse using those genetically engineered methods to create exactly the mutation she wanted to make, to learn something fundamentally new. Now, each of these young women at the beginning of their careers went on to make very distinguished contributions in developmental biology. So Liz's work has taught us how the left and the right sides of the embryo become different from one another, and how the extra-embryonic tissues of the embryo communicate with the embryo itself to guide its development. Gail has defined the ways that cells within an embryo communicate with each other to organize themselves, to teach themselves how to make complex organs like brains and limbs and hair and teeth. And these insights that we have now are just a few of the insights that we have about how the embryo works and how development works, how mouse development works, and how our own human development works, that have all been made possible by embryonic stem cells. So in the popular press now, not just in science but in the world, we talk now about using stem cell research to make cells that will rescue a human with a disease. And when we do that, we are building from the work that these three extraordinary women did to generate embryonic stem cells. Because of their work, genetically altered mice are now the method of choice for studying human development and disease. And every university, every biological problem, every department has scientists who use these methods to try to understand biology and medicine. And every university has these miraculous mice on the hoof, as we say, that trace their heritage from a cell in a dish and not from a mother or a father. So I would like to thank these women for what they have taught us and invite Gail Martin, Liz Robertson, and Paul Nurse to the stage.